Hey there, everyone. We'll go ahead and let folks start coming into the Zoom room. I hope everyone is having a fabulous day so far and looking forward to some good Thanksgiving celebrations if that's what you're up to next week. Go ahead and navigate to the chat box at the bottom of your screen, your Zoom window, and let us know if you've joined us for a Vernier webinar before. We would love to hear from you. And as you're typing into that Zoom box, make sure that it's set to send to everyone. That just helps us make sure that your message is seen by everyone, so that as you're asking questions and we're answering them, we can reference something that everyone can see. And it looks like Dana is joining us for the first time. Welcome. Thank you so much. Julie, fabulous. Thank you for coming. Oh, and another first timer, Holly. Hello. The fabulous news is that a lot of this is open to questions. So as we're going through this presentation, I know that our amazing presenters would love to answer your questions. So go ahead and drop your questions in that chat. We'll help you in chat or we'll, answer it verbally. So make sure that, you're using that feature. We are here for you. Oh, it looks like we have another first timer, Jennifer. Thank you so much for coming back. All right. Well, I'm sure we still have some more folks coming in, but in the effort to kind of make the most of our time together today, let's go ahead and hop into some housekeeping items. Do you mind going to the next slide, Colleen? Fabulous. So just two important things to note. You will receive a recording of this webinar. It typically takes about 3 to 5 business days to get it all edited and situated. As soon as it's ready to go, we'll put it on our website and our YouTube channel. You will have access to this presentation, and both the website and YouTube channel there are linked, so you can easily get there. We'll also send a follow-up email with the resources that I will share with you today, as well as that recording. So keep an eye on your inbox that is coming your way. And as I was just mentioning, we would love to answer your questions during today's session. So please use that chat section. Make sure you're sending that message to everyone so we can all see, and we will have our eye on that chat box. If we happen to not get to your question or we kind of want to speak with you further and troubleshoot with you one-on-one, go ahead and reach out to us at biology@vernier.com. And with all that being said, I will pass it off to our amazing presenters. Thank you again for joining us.
Good morning or afternoon, everybody, depending on where you're joining us from. Just to kind of go over what we're going to be talking today. So just give you a little background on me and Sara. Talk about what inquiry-based learning is, and then we're going to do an activity looking at testing enzyme activity, and then have a little Q&A or discussion afterwards. Open. And the big thing is make sure you ask any questions at any time. We do have the resources available in the folder for you, so you will have access to this presentation along with copies of the lab that I'm going to be doing as well. And again, like Dylan said, make sure when you ask questions to have that available for everyone to see, just so we make sure if people sometimes have a lot of the same questions. That way we can get them all covered at once. Okay, so a little bit about me. My name's Colleen McDaniel. I have been at Vernier now for 10 years. I have a bachelor's in science of biology from Truman State University, middle of nowhere, Midwest Missouri. And then I got my master's of science in science education from Montana State University in Bozeman, which was an amazing, awesome program there. Actually, before I got my master's, I was a research scientist. I was doing fruit fly research, looking at molecular and human genetics, and then was lucky enough to get a grant where I became certified as a high school teacher, where I taught for about 6 years in Houston, Texas, where I taught biology, pre-AP biology, and AP environmental science. So that's a little about me, and here's Sara.
Hi, my name is Sara Tallarovic. I have a PhD in zoology from Oregon State University. I also have an undergraduate degree in biology from Northern Arizona University. I was a professor of biology for about 15 years. I taught biology in Texas, both Austin and San Antonio. And my background is in animal physiology and behavior, as well as neuroscience and endocrinology. So happy to be with you today.
So let's talk about what inquiry is. And for me, inquiry, I think, is something that's really important that we discuss and make sure we understand, because when I entered the classroom, this was the big hot button item when I started teaching was inquiry is the best. Inquiry is amazing. Do inquiry all of the time. And it was kind of overwhelming, honestly, to especially as a high school teacher, thinking about being able to do inquiry with my students. And it was a matter of me really digging down and understanding exactly the goals of what an inquiry-based lab instruction is. So for me, the way I look at inquiry is it's really a method of doing some labs that allows students to explore more and ask their own questions more. And what's really great about this method is it's a really a lot more true to what we would talk about with the scientific method, is that it encouraged the students to ask questions, make observations, make an experiment, and then come up with some conclusions on their own. And the key that I found out the hard way is that inquiry done right is very not unhinged. When I first did my very first inquiry experiment with my AP students, I was doing a lethal dose lab with brine shrimp. And I told my students, okay, so we looked at salt water and the effect it has on shrimp. Now we're going to do inquiry where you're going to ask a question and figure out something you want to find a lethal dose of. And it just went off the rails. The students were all over the place of like, oh, I want to test this. I want to test this. I want to test this. And I had no real way of kind of reining them in on that. And so the students just brought in things to test, and we're testing like Axe body spray, where it's like, okay, well, that's an aerosol. How are we going to get it out of the bottle and make it a liquid and have it a measurable amount? And it was that kind of thing that made me realize, okay, I need to step back and reevaluate exactly how the best way to do inquiry is. And so I kind of came up with these little tips and tricks for inquiry that a lot of times people make it seem like if you're not doing inquiry, you're not doing good labs. And that's not the case. There are some lab activities that are really tuned in and great for doing inquiry. And then there's other ones that are not the best situation to do an inquiry type lab. Labs that I find are really key to do a good inquiry are going to be labs that have lots of different variables that you are able to change and able to change them easily. And that's kind of the key here, is making something that's going to be a lot simpler, and so that the students don't get bogged down in figuring out a procedure, but are able to spend a lot more time thinking about their questions and the results that are ending up from their tests. The other huge advice I have is start with an introductory activity. So you want to start with an activity that's going to introduce the students to kind of the concept they want to look at. One, it gives them background on that concept. Two, it gives them experience with whatever equipment they're going to be using to be able to do the experiment. And it kind of helps them see how a good procedure is laid out that will allow them to kind of come up with their own procedure to develop. It also is nice to do this kind of introductory activity to set a basis for questions, or what I also call kind of that wonder factor of, okay, well, X happened. I wonder what would happen if I changed A or B, and see how that is going to affect the results. The other thing is be prepared to give guidance. There's going to be, depending on the level of students you teach, anywhere from freshmen in high school to freshmen in college, you're going to have some students that are going to be really comfortable with this, and probably the majority really uncomfortable with this, because they've been so used to essentially being handheld, of being told how to do a lab activity. So you want to make sure, what I always suggest is have a couple variables in mind that you want to encourage students to try testing out. Or even do a guided type inquiry where you start with your initial experiment And then you just assign inquiry activities as students kind of come up with them through a classroom discussion. And the other key thing to keep it from going unhinged is to kind of set limits, but encourage exploration. Make sure you have students kind of run their ideas by you before they just set out ahead and start doing the experiment. You want to make sure that they understand the limitations of research. And that's a big thing with science too, is that not all questions are going to be easily answerable. That's why we still have questions out there that we're researching for science and trying to understand. So you want to encourage that exploration, but kind of make them think about, well, okay, maybe that isn't a great idea. Like for my students that were bent on doing Axe body spray with the brine shrimp, what I should have done is like, okay, well, this is an aerosol. You need a liquid. So how are you going to get that out? Maybe we should look into something like Axe body wash instead, and kind of try to guide them to other choices to help them see what's going to be a good variable for them to actually test that's doable. Any questions so far? Nothing.
Colleen, I would add from personal experience as well to make sure that students understand that if they manipulate more than one variable at a time, they confound their results. So unless they can do so many replicates to make up for the fact that they've added extra variables, they get so excited, I think, when they really get rolling on inquiry, that they often want to manipulate too many things at once. And so getting them to focus on a single variable is one of the best things you can do, and then have them do subsequent studies if they really get rolling.
Yeah, that's exactly a good point, Sarah. Because I think a lot of times a good thing with inquiry is also helping students understand what variables are and why you want to change only one variable at a time, or be careful of what one you manipulate, because it can have those compounding effects on other variables. All right, so what is a good inquiry experiment? Well, one of my favorite experiments to do that I find super simple to do an inquiry activity with is looking at enzyme activity. And I really do think this is kind of like what I would consider the poster child of a great inquiry experiment. Mostly with doing the initial setup with yeast and using a gas pressure sensor. It's super quick. It's a very simple setup. Students can get results in under 3 minutes, which for a biology lab is pretty impressive, I have to say. Materials are going to be really easy to come by. There's lots of different variables that students have the ability to alter. And if you are doing a sensor-based experiment, there's actually a lot of sensors that you can use depending on the type of setup for it. So for example, with this lab setup, you can do an experiment looking at gas pressure. So looking at the gas pressure being released, or the gas being released and how that increases pressure. You can look at specifically the oxygen gas concentration that is being produced by the reaction. And if you're doing AP biology, for example, you can use a spectrophotometer and use guaiacol as a color indicator of the reaction taking place. So let me change my screen sharing here and show you my lab setup. So I'm going to confirm, Sarah, you can see my new screen sharing. Fantastic. So one of the reasons I like this enzyme lab is, like I said, because it's so easy and quick to set up. So what I have is I have yeast as the source of my enzyme. So the yeast are going to be producing my peroxidase, and then I have peroxide and water. And I'm basically going to create a situation where I have 1.5% peroxide that is going to be reacted with yeast. So first thing I need to do is connect my gas pressure sensor. So I'm using the Go Direct one. I'm gonna turn on and connect. Oops, not a temperature probe. The problem with being in the Vernier office. One too many sensors available for us to connect to. There we go. Right, and then I'm going to change my sampling rate down here. Right now, we have it as 1 sample per second over 900 seconds. So I'm going to change this duration from 900 seconds to 180 seconds, because it doesn't have to be a super long data collection for this. Click done. Okay, and then I'm going to set up my experiment. How I like to have this set up is I do 2 test tubes, one labeled for enzyme, one labeled for substrate. And what I do is go ahead and add 1.5 milliliters of peroxide to this test tube, using a graduated pipette to make life easy for me. And then in the enzyme tube, I'm going to add 1.5 milliliters of water. And so this essentially is going to set up a dilution where my substrate is 1.5% instead of 3%. And then for my enzyme, you can't see it, it's off screen here, but I have a beaker of yeast that I started growing last night. It's 1 packet of baker's yeast to 100 milliliters of water with a little bit of sucrose in it to kind of get them happy and bubbling. And I have them on a stir station so that I want that yeast colony to constantly be stirring and have a good homogeneous mixture going. So I make sure I'm grabbing a good amount of yeast. The other trick that I did that's really important, especially if you're using any enzyme actually to do this type of experiment, is you want to run a test ahead of time so that you know exactly how active this enzyme is before you hand it off to the students to have way with. So for example, about half an hour ago, I tested this with 2 drops of yeast and found that the reaction rate was there, but it wasn't really fast. So instead of 2 drops, I'm going to do this experiment with 3 drops of yeast. So I'm going to take pipette, take my yeast sample from the center of my beaker that it's growing in, and then I'm going to add 3 drops of yeast to my enzyme. 1, 2, 3. And since I set these up in 2 different test tubes, what's nice is I don't have to rush and hurry to get them combined in order to do this experiment. So I'm just going to kind of take my time, going to give this a little swish, and then I'm going to pour the yeast into my enzyme, into my substrate, give it a little swish, put my gas pressure sensor on here, and start data collection. So very quick setup, very simple. And you can see I'm already getting some great activity taking place here. So I can see that I have my gas production, my pressure is increasing, which shows that gas is being produced and increasing inside of my test tube there. And so during this, students can start to make some observations. If I were to look at this test tube closely, I could see that there's bubbles starting to form. So I can look at then the reaction that's taking place, write out the reaction and figure out what exactly is in those bubbles. And that's where some people might want to use an oxygen gas sensor instead of a gas pressure, because that can actually measure the actual gas that's going to be coming out there directly. A couple variations on this: instead of using test tubes, if you're using an oxygen gas sensor, you can use a larger flask. Usually we recommend using the Nalgene bottle that the O2 gas sensor comes with. And with that, we also recommend using a stir bar at the bottom because you want to make sure that you're distributing the oxygen gas up to the sensor so that it can actually be sensing the increase in concentration. And then you don't get these like blips of all of a sudden there's like a burst of oxygen, a burst of oxygen, a burst of oxygen kind of thing going on. So I set this up to do 180 seconds. Looks like I don't even have to do it for that long. And so as the students are doing this, the other thing to encourage them is start asking questions now. So what could affect my enzyme rate here? What exactly is happening? So I have my yeast that are my enzyme that are breaking down this hydrogen peroxide into water and oxygen gas. How can I affect that rate? What are different things that I could adjust or modify in order to get a change in my reaction rate? And it looks like my pressure went too high. So I'm going to go ahead and stop data collection and I'm going to go here and zoom to all of my data. And you can see once I reached a certain point, my stopper on my reaction popped off there. So we're going to ignore this end part here. And what I can do is just highlight a portion of my graph. And then I can look at my curve fit, do a linear fit, and look at the rate of increase. So I can see that my pressure was increasing by 0.27 kilopascals per second. So that's going to be our baseline. And then from there, students can now adjust a variable and see how that affects based off that baseline and doing a comparison. And what's great about this experiment is that it happens so quickly. Like this was just over 2 minutes of data, that it can be really easy for them to run multiple trials, which actually helps them kind of do better in that inquiry aspect because they can change the variable multiple times and see what kind of effect that has on it. Questions on that experiment?
Not a question, but some of our participants suggested temperature, pH, substrate, concentration, all as items that could be manipulated.
Fantastic. So going back to the presentation here, you all read my mind. So the great thing with this enzyme activity is there's tons of different variables that you can alter and test with it. One of the two easiest ones are going to be your enzyme and your substrate concentrations. So for this case, I used yeast. So we don't know the exact enzyme concentration of the yeast, but what we do know is how many drops I put in. So I could adjust the number of drops, and that would essentially adjust my enzyme concentration. If you're teaching a little more of an advanced class or want to be more precise, you can actually purchase purified catalase from Flynn, and I'm sure other sources as well. And the nice thing about the catalase is that it actually has the amount of enzymatic units per milligram. So you can actually have a very quantitative amount of enzyme that students are adding. The only downfall of catalase is it's not very stable. So this will go bad very quickly. So it's something you don't want to purchase until you're getting ready to do that experiment. With substrate concentration, I was just using literally grocery store hydrogen peroxide. You can get up to 6%. I wouldn't recommend really going higher than that because then you get way too high of an oxidizer going on there. But you can allow students to then vary that substrate concentration and see how that affects enzyme activity. The other tip with this is whatever peroxide you're using, you want to use fresh because peroxide does degrade over time. So the fresher that peroxide you have, the better results you're going to get. Temperature is something you can also manipulate. You can have students play with the temperature of the substrate or temperature of the enzyme. You want to make sure that they don't do both at the same time, because again, you don't know which factor the temperature is affecting. Is it affecting the substrate, or is it affecting the enzyme? Or we can look at just general, how is it affecting the reaction itself? One of the ones that at first when I read it, I was very intimidated by, but then was like, oh, this actually isn't that hard, is adjusting the pH of the reaction. So in this reaction, I just used distilled water to dilute my peroxide. What I could do is, instead of distilled water, I could use a pH 4 buffer or a pH 5 buffer, or I basically just change out that water for a different pH buffer. And that allows me to then look at the effects of different pH on that. And the guaiacol lab, doing the turn-up peroxidase with guaiacol is one of the best ways to do the pH testing. It gives such beautiful results. So much so, the first time I did it with the spectrophotometer, I thought I messed up because it was too perfect. So it's one of those labs that I highly recommend doing, just because it works so well. And that kind of comes also then to our source of enzyme. So the cool thing about this enzyme lab looking at peroxidase is that there are different sources of this enzyme. And it's mostly because all cells produce hydrogen peroxide as a waste product, and it is toxic. So we have to make sure our bodies have ways to break this down in all living organisms. So you can look at a source of the enzyme and compare it between different types of plants. For example, potatoes, that's probably one of the number one that people often use for a source of this enzyme is potatoes, compared to rutabagas, compared to turnips, compared to any other vegetable that you can get. I have found the most stable enzyme I have ever used, period, is from turnips. It's amazing. That's the one I used for the guaiacol lab. And I had that enzyme prepared and then used it again 2 months later, and it still gave me a reaction. It was really impressive. You can look at animal sources of enzyme. The catalase comes from Flynn. That is a bovine source. Liver, that's our usually most disgusting #1 thing that we use in the classrooms. And again, though, it's not super stable. So it's one of those things you have to keep in mind. You have to make sure it stays on ice. You might need to replenish it throughout the day if you have multiple class periods throughout the day doing this activity, because the enzyme activity can degrade over time. And then the one I use today is yeast. So this was just baker's yeast, but you could test different things, such as different varieties of yeast, because yeast has all sorts of strains that are available. You can compare brewer's yeast to different types of wine yeast, different types of brewer's yeast, fast action yeast versus normal yeast for baking. It opens up a whole world of availability of options for it. So that's why it's really easy for this lab activity for students to kind of go off on different pathways to do these sorts of investigations of inquiry. And the procedure is very simple, very easy for them to manipulate and change, and have really good success at it.
Colleen, we have a couple great questions. We had a couple different people ask about pH. First question was, what did you use for pH buffer? And the second question, which is related, I believe, is that how do you dilute the pH 4 buffer to make other pHs?
Okay, so you don't want to dilute different pH buffers. So pH buffers are going to come in as set pHs that have been basically, I don't want to say calibrated, but made to specific specifications to be that one pH. The easiest thing I find is buying pre-made packets that you can find on Flynn, basically packets that you would use to make a pH buffer to calibrate a pH probe. And what's great is these packets are already optimized for that pH, and usually it's one packet or one capsule to 100 milliliters of distilled water, and your pH is set. You don't have to do any adjustments as if you were making a pH buffer on your own, which anybody who has done research know, in any molecular research, making a pH buffer on your own can take hours. It is not a fun thing to do unless you really enjoy that kind of thing. So I know, for example, we sell buffer capsules that we use for calibrating our pH. Oftentimes, I'll just use those to change up and use like a pH 4 buffer, a pH 7, and a pH 10. Get a big spectrum of looking at the different pHs. The Flynn has a turnip peroxidase kit that they sell for the Guayaquil lab. That comes with pH packets so that students can adjust that variable as well.
If you do want to get fancy, you can have your students mix phosphate buffers. But yeah, I would make sure you plan plenty of time for them to be blending the monobasic and dibasic buffers together to get the target pH that they want.
Yeah, and that would be something I would say is definitely AP or college level.
Yeah, and we do have our lab. I know that some of our labs that have students mixing the buffers, we do have some chemical formulas listed for you in the teacher notes.
That we do, yes. Any other questions?
We did have an analysis question that so somebody asked about the root square mean error, which I put that in. They asked about RSME, and then they also asked, why do students get negative values for R?
That's a very good question. The negative for R throws me off. Have you seen a negative value for R, Sarah?
I have not, like as in a negative, that's what I wasn't sure about. If
they
if
can
we
get a negative slope, not necessarily for this reaction, but a negative slope would show things are decreasing, not increasing. But I can't say if I've seen a negative R value.
Yeah, that usually indicates that there's no correlation between the variables.
Yeah.
So usually the reasons that you would get, it's usually a low value for R is if the line fit is not a good fit for your line. So if they don't have a particularly strong relationship.
Yeah.
At least one of my groups will get negative 0.998. That's interesting. You can always send us data. Send us a data file and we can always take a look at it and see if we can figure out what may have happened with the lab prep.
Yeah, that's the best. If you have students that get some odd data, save that file and email it to us, biology@vernier.com. We can definitely take a look at it and say, oh, okay, I think something went wrong here. It's one of the easiest ways for us to be able to help diagnose an issue that might have occurred. Any other?
I know we had a few folks that wanted to know more about the Guayaquil lab, but you did spend some time talking about it. So if that, we can ask our audience if you want to hear more about that one. Question, is RSME a better indicator of fit? Better, do you mean better indicator than R? That's a good question. It's usually root mean square deviation or error is usually basically it's looking at closely related measures and the difference between a relationship versus a predicted relationship. So sort of how well does it fit the model, for instance? How well does it fit the line curve? So either one can be used as an indicator of whether of how good of a fit it is to the model that you're applying to it. And we did get a couple of yeses on, there are folks who would love to hear more about the Guaiacol lab.
All right, fantastic. Sadly, I was not able to get all of the pieces in order to do that lab because of traveling for various conferences and shows over the past few weeks. But it's one of those, Flynn has an awesome kit that we actually have written the written lab instructions for and how to use that kit with our spectrophotometer. It's a little bit more time consuming prep wise than the one we did today. And that's purely because you do have to make a turnip peroxidase enzyme. And how you make it is it's literally you blend a certain quantity of turnip with a mixture of monobasic and dibasic buffers, which ends up being about a pH 5 buffer solution that you're going to make it in. Then once you've kind of pulverized it and blended it, you have to strain it through some cheesecloth to get rid of all the fibrous matter out of it. And then it's ready to go. And it's a matter of mixing the turnip peroxidase with a little bit of peroxide and then guaiacol and buffer. And what I would do is mix them in a test tube like I did this way, where I have an enzyme test tube and a substrate test tube. When I add them together, it mixes, and then I pour it directly in the spectrophotometer into the cuvette, and then put that right in the spectrophotometer because the reaction happens very, very, very quickly usually. And what's great is on the spectrophotometer, we tell you what wavelength you want to measure it at. And you're going to do a time-based data collection. So the students will see that the absorbance changes over time and that it's going to increase because the sample's getting darker. And that's going to give you your reaction rate. So it's going to be all based on how, on the, how dark that sample gets in a period of time. There was a great little article I wrote about that about, gosh, 6 years ago, I think, maybe. Let me see if I can find that. Here. Oops.
I'm also helping search, Colleen. If I can find it, I will drop it into the chat. It may have been on the older version of our website.
I found it recently on the new one, so let me here. See what I can find.
Was it from 2014?
Yep, I found it.
Got it.
Got it here. So what's nice with this, I just went ahead and copied and pasted it right into the chat there. This is an activity that can also use a colorimeter as well. So if you don't have a spectrophotometer, one of the wavelengths on the colorimeter will work as well. Let me show my screen here. Should switch. There we go. Should be showing. Is that my screen showing now?
Yes.
Okay, perfect. So this article I wrote, gosh, almost 10 years ago. Time goes fast. So this is gives you a great picture here showing how I changed the enzyme concentration, and we're doing measurements around 500 nanometers. And like I said, you can use a SpectroVis Plus or a colorimeter to do this lab. And you can download here. I have there's student instructions and the teacher information located there. I added some updated instructions are in the resource folder for this webinar as well. That I made sure that when we rewrote the Biology with Inquiry book, that we made sure to incorporate this activity into that book as well. And like I said, it's one of those activities and what's great with that kit from Carolina is there are some things that are that will run out over time, but honestly, the amount of guaiacol you get is enough to do this lab probably for 4 years because you only need a very small portion of guaiacol. And then afterwards, what I recommend doing is just purchasing mono and dibasic solutions and the buffer packets to make the different pH buffers for you. Any other questions?
We had a great question that I think is worth sharing with everyone. I answered it in the chat, but we had a great question about how often do we update our lab manuals? And the question that I offered, or the answer that I offered was typically when we do a major revamp of a book, it is because we've released a new software title. So it doesn't happen super often. And if we release new sensors, a lot of times those labs may be freebies on the website. Sometimes we will also do completely new book titles when we have a number of new products available, or a lot of new lab ideas that we'd like to put into a new collection of labs. But the updating usually is for the purpose of adding a whole new set of protocols for a new software. So for instance, when graphical analysis came on the scene, we did revisions of a lot of our lab books so that there are instructions for using graphical analysis with those lab preparations from that book. Not quite all of our books have been revised with that, but all of our main titles have.
And kind of on the same note, if we find a flaw with one of the experiments, we will get those updated as well. Now, we don't necessarily update the printed version of the book all the time, but that's where having an online account on the Vernier website gives you access to the most recent titles, recent updates of the titles. And any physics, yes, there are, they have just working on finishing up all the advanced physics books to get them working with graphical analysis right now. And yesterday we just released forensics with our new forensics book, forensic chemistry
with Vernier.
Thank you. Also just got released as well. But what's great is once you've purchased the book, so for example, if you already own Biology with Inquiry, you have access to those updated files, even if you purchased the book 10 years ago. It's a site license, so once you have it, it's there. So if you bought Biology with Vernier 10 years ago, or sorry, investigating Biology with Inquiry 10 years ago, the guaiacol lab was not in that book, but I've now updated it and that lab is now included. So you just have to go download it from our website under your account now, which is really convenient and helpful. The original lab, the new probes work with these computers. Do I need it? Excellent question, Holly. So the Go Direct sensor I used today, that actually does not even need an interface. It's going to connect directly to a computer or a Chromebook. It won't connect directly to the original LabQuest, but it will work with a LabQuest 2 or higher. But the advantage of it is that it doesn't have to have that interface anymore. That's kind of why the Go Direct is very convenient, especially if you have some of those older interfaces. But if you have older interfaces and they still work, awesome. That's fantastic. And if there's any questions on things that do or don't work, don't hesitate to reach out to us. That's what we're here for. And I'll go to the next slide here for the contact us. We are available to help you. That's what our jobs are. We want to make sure that if you're running this enzyme lab and getting weird results or things that are not right, call us up. We'll talk through it. We are more than happy to help you out. There's a customer that was doing the guaiacol lab that I talked through that lab over a period of 2 months to make sure he was able to do it successfully with his IB students. So it's one of those, that's what we're here for. You can call us, ask for somebody in biology. You can email us at biology@vernier.com. And during our office hours, we also have chat available as well. So you can actually just pop in and chat with somebody that's available on the website. And the chat's only available when we are around. So if we're not around, the chat's not going to be on. So we are closed on weekends, sadly. Not sadly for us, but but that's where email can be great, and we'll get back to you as soon as we can. So awesome. Well, thank you everybody for joining us. I hope I gave some good ideas for inquiry and kind of help people feel a little more comfortable with it, because I know when I started doing it, I was not comfortable with it and thought I was crazy for even thinking of being able to do this with my students. So thank you all again, and have a wonderful rest of your week, and happy Thanksgiving next week.
And don't forget to give us feedback on the webinar. Dylan has been putting the
Yes.
link in the chat.
Thanks, everyone.
Thank you.
Yes. Bye, all.